Hi, it's Mr. Cotton. We are working on somatic cell cycle. That includes mitosis today. If we were trying to make gametes, then that's a different cell cycle. So the gametic cell cycle would be including meiosis. We'll do that later on with Mendelian genetics. I think Acelius is teaching that one. And so somatic cell cycle, and then DNA structure and function. So hope you like it. We'll start off by looking at a picture that I have over here. And this is the cell cycle. And in this picture, it's a circle, so there's not really a beginning and an end, but like cells just split here. So this is the beginning. You kind of have a baby cell, so to speak. And in G1, it stands for gap. G1 is gap 1 and G2 is gap 2. But what happens is growth. So I always think of the G as meaning growth. The cell just gets physically larger. And that makes sense because we just split into new cells. So they're kind of like, again, like baby cells, so to speak. So in G1, we grow. And then the thing that we want to consider is eventually we're wanting to make two identical cells. So if we're doing a cell cycle that includes mitosis, we're wanting to basically make exact copies. It's like a Xerox copy machine. Meiosis is different. There you want cells to be different. You want genetic diversity. Here you don't. You want copies. So if I start with one cell and it eventually is going to be two identical cells, then I kind of have to get bigger. If I'm going to split, I have to get bigger. Then I have to copy all the crap inside. So the S phase is when we synthesize DNA. We replicate DNA. A replica is a copy, like a replica of the Mona Lisa looks just like her, but it's not worth very much. And so DNA replication happens in the S phase, the synthesis phase. Then in G2, what else would we need copies of before we split? Other organelles and things like that. So now we start kind of copying the other stuff along the way. I know it's mentioning some of these checking things right here, but we're going to need more Golgi and ER and stuff like that. Then in mitosis, we break it down. Typically, we break it down into 4 stages. You can break it down into 5. So we typically say prophase, metaphase, anaphase, telophase. If you read a different book or something like that, they might have a prophase, prometa, and then metaphase. So, but we'll say prophase, metaphase, anaphase, telophase. And then that's mitosis. And then cytokinesis is when the cyto splits. So common things that are misconceptions or that students miss on the test. It seems like the STAAR test cares a whole lot about the S phase. In fact, I don't recall, I'm sure it's happened, but I don't recall them asking about G1 or G2. But I recall them asking this probably 2 out of every 3 or 3 out of every 4 years. And then they usually ask a mitosis question too. Okay, so the way that we want to look at this is, let's say that I've got a normal cell and I have a chromosome 1 from dad and a chromosome 1 from mom. And I eventually want to make identical copies. So I'll start off in G1, chromosome 1 from dad, chromosome 1 from mom. These are not identical because your mom and dad are not identical, right? That's weird. So these, it probably won't come up, but they're called homologous chromosomes. That's probably not going to be on the STAAR test. But then as we get through to the S phase, then we make copies. These copies you might have heard are called chromatids or sister chromatids. They should be identical to each other, and they're joined at a centromere in the middle. That kind of holds them together. Then after that, we replicate some organelles, and now we start going into mitosis. Well, in mitosis, we've got this picture right here. I'll kind of take you through it. I think it's a little bit small. So essentially what we want to do is in prophase, we want to kind of prepare to get everything ready. So I get all my chromosomes condensed down and I start dissolving away the nucleus and things like that. These guys right here are called spindle fibers. And their job is they're supposed to reach out and grab the chromosomes and help pull them apart. So the idea is this: chromosome from dad, chromosome from mom. I go through the S phase. After the S phase, these guys are partnered up. So if I could, in prophase, get a hold of the chromosomes, and then in metaphase, line them up in the middle, and then in anaphase, pull them apart. Anaphase, 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 anaphase. I'm done pulling them apart. Telophase. Then I could cut the cell down the middle, cytokinesis. So the problem is these things right here that make the spindle fibers, they're called centrosomes, they can't get to the chromosomes because the chromosomes are inside the nucleus. So those spindle fibers can't grab them. So spindle fibers appear, but then the nucleus is going to have to dissolve. The nucleus dissolves. Now those spindle fibers can grab. And if we can line everybody up like kind of, in a line like this, then since they're already partnered up with their identical copies, if I just rip them apart, my right hand and my left hand should end up with the same stuff in them. So that's what's going to happen. Metaphase, they line up in the middle. Anaphase, we're pulling apart. Telophase, we're done pulling apart. And then cytokinesis, we cut it right down the middle. There are little things like a little hand thing where it's like, here's prophase. We're starting to dissolve away the nucleus. Here's metaphase. They're lined up in the middle. Here's anaphase and telophase. So it's like prophase, metaphase, anaphase, 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 telophase. And then if we cut the cell down the middle, cytokinesis. Cytokinesis is not a part of mitosis. It's after mitosis. In mitosis, you're just trying to separate the genetic material. You're trying to get identical copies going to each side. Then you split the cell, the cyto. So take just a second and see if you can figure out which of these pictures, I'll shut the lights off for just a second. See if you can figure out which of these pictures is depicting which phase. So where is metaphase? Good job. Metaphase, everybody's lined up in the middle. What about anaphase? Good job again. Anaphase, I'm pulling the chromosomes apart. Here's prophase. I'm just now kind of dissolving the nucleus. My red spindle fibers are getting ready to grab the chromosomes. And then here's telophase. You can see that I've now got my DNA totally separated. I'm rebuilding a nucleus. And then I'm even starting cytokinesis here. All right. The last thing before we hop into some questions. There are quality control checkpoints along the way. So just realize cells aren't just going through this cell cycle as fast as possible. Yeah, if I get a cut on my arm, then now on either side of the damage, I hope that my cells divide quickly to kind of fill in the gap. But cells aren't just always going as fast as they can. So I have quality control checkpoints. But sometimes cells misbehave and they ignore those checkpoints. And if cells are growing out of control, what do we call that? That's right, cancer. So cancer is anytime cells are growing out of control. So what does kidney cancer have in common with liver cancer, with lung cancer, whatever? It's cells growing out of control. So we are supposed to have controlled cell division. All right. So now let's go over here and try out a few questions. Just like with my last video, each teacher that does these might do them slightly different, but I usually go through about 4 or 5 questions with you, and then I'll have a couple of extra. So when you guys go to fill out that Google form, there'll be a few unfamiliar questions on there, but most of them we'll actually go over right here. Scientists can bioengineer skin in a lab to treat severe burns and other types of skin injuries. This bioengineered tissue is grown from living cells. The cellular process that enables the cells to grow and develop into tissues is conjugation, meiosis, budding, mitosis. So what do you think? It's mitosis. If we were making germ cells, gamete cells, sperm and eggs, then it'd be meiosis. Conjugation, that's something that bacteria do, and budding is something that yeast does. Fungus. All right, next up. This one I'll read to you. Well, I guess I read all of them to you, but I know this print might be a little bit small. This model represents the change in the DNA content of a cell during the cell cycle. Which part of the model represents the S phase? So don't forget to read your X and Y axes. Cell cycle phase, so there's like phase 1, 2, 3, and 4. All right. Complete sets of DNA in the nucleus. So I've got a certain amount of DNA, and all of a sudden it looks like I'm doubling it. Now I have twice as much. This is the process of me doubling the amount of DNA. So which part of the model represents the S phase? It's this part, part 2. And that's because if we go back and look, the S phase I guess I explained it on this diagram. The S phase is whenever we copied the DNA. So that's when our amount of DNA would be increasing, when we're copying it. So that makes sense. Okay, next up. The diagram below represents the cell cycle. All right, when cells leave the cell cycle, they exit during G1 and then enter G0. And you're like, G0? Crap. Where were you on that one, Cotton? A resting period. But you're going to have enough information. They're going to tell you about G0. Most normal cells can leave G0 phase and re-enter the cell cycle at G1 before entering the S phase. Cancer cells are different because they cannot enter G0, and they are likely to do which of the following? So all of a sudden you look at G0 and you're like, we didn't really talk about G0 in here. So what am I going to do? It doesn't matter. Cancer cells are different. They can't enter G0, and they are likely to do which of the following? Fail to complete the S phase, mutate during G1 phase, repeat the cell cycle continuously, die after completing mitosis. Well, again, it comes down to what's cancer? Cells growing out of control. Fail to complete the S phase, well, that's not growing out of control. That would be like death. Mutate during G1, you're kind of like, I don't know, mutations can cause cancer, but just because there's a mutation during the G1 phase wouldn't necessarily mean that it's cancer. I can have a mutation, and that means that instead of having brown eyes, I have blue eyes. It doesn't have to be cancer. Repeat the cell cycle continuously, that's correct. If I just keep going continuously, that's cancer. And then die after completing mitosis, that's the opposite. Cancer is growing out of control. Death is, the opposite. All right, next up. Telophase is a stage of a cellular process that begins after the chromosomes have moved to opposite poles of the cell. During which cellular process does telophase occur? So I just always think of PMAT. Mitosis contains PMAT, prophase, metaphase, anaphase, telophase. So it's not translation. That's like genetics when we convert RNA to DNA or RNA to protein. It's mitosis. Interphase, by the way. If we go back and look at this diagram, they don't show it on this diagram, but interphase is everything here. So G1, S, and G2 together, they can also refer to all of that as interphase. So interphase is a word, and it is like at least related to this unit, but it's still not the right answer. So telophase happens in mitosis. In meiosis, it'd be like prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, like that. All right. Last one, and then we'll leave the rest for you to work on, and then we'll go on to DNA. Cells pass through a G2 checkpoint before entering mitosis. Okay. Ideally, if DNA damage is detected, the cells do not enter mitosis until the damage is repaired. Why is DNA damage repaired before cells enter mitosis? Okay, so that another round of DNA synthesis does not have to take place, so that the chromosomes can align at the metaphase plate during mitosis, so that the cytoplasm can be divided equally between the two daughter cells, so that the healthy daughter cells are produced, allowing the organism to continue growing. So if we go back and look at our checkpoints here, before I move on to mitosis, if I find out at this checkpoint that there's some DNA damage, if I just let it go anyway, then the mutations get passed along, right? So hopefully, if I notice there's a problem, I fix it. The whole point of quality control. If I'm building cars and I realize there's something wrong, I don't just say, send it along anyway, I should fix it. So we do that. That way, future generations aren't screwed. So another round of DNA synthesis does not have to take place. Well, you should only have one round anyway. So that's no good. So that the chromosomes can align at the metaphase plate during mitosis, well, they would still align anyway, whether there was a problem or not, whether there's a mutation or not. So that the cytoplasm can be divided equally. No, that's also, again, if there was an error in my DNA, I might still be able to do cytokinesis just fine. The reason that we want to repair it is so that healthy daughter cells are produced, allowing the organism to continue growing. All right, let me pause for a second. All right, time for a little bit of DNA structure. So with DNA structure, keep in mind that DNA and RNA are the genetic material, and we had that picture up here previously of our nucleus. DNA is inside the nucleus. DNA usually gets wrecked if it's outside the nucleus. There are DNase enzymes that'll destroy it, but RNA can safely leave. So we're going to focus on the structure of DNA. So in DNA, we've got 4 bases that comprise DNA, and then they're put together in a sugar phosphate backbone along the way. So right here, let's look at this picture. Here's a picture of DNA, and it's kind of got everything that we need just in this one picture. So here's a phosphate group, a sugar, and a base. Phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. These bases do have a lot of nitrogens, so your teacher might have referred to them as nitrogen-rich bases or nitrogenous bases or something like that. At this point, you probably don't know like why emphasize that there's nitrogen in it after you've had chemistry. Most students take chemistry after bio in Texas. So after you've had chemistry, then you'll understand why would we care? Why would we point out that they're nitrogen-rich bases? That's kind of unique. So same thing on this side, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. So a monomer is a single part or a single piece. Mer is piece. Mono means one. A polymer is many of those pieces. So anytime you have a repeatable pattern, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, then that is a polymer. And the monomer is the repeated part. So like 1, 2, 3. Let me show you what I mean. If I go over here and I write on the board, one, two, three, 1, 2, 3, 1, 2, 3. What's next? 1, 2, 3. It's a repeatable, recognizable pattern. So the single repeatable part is the monomer. I know that that's actually 1, 2, 3. I know that that's 3 different numbers, but that functions as a monomer because this is the single repeatable part. If it was 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, then 1, 2 would be the monomer. So now what we want to do is look at our DNA and see what's the monomer. Phosphate sugar base, phosphate sugar base. So the monomer is a phosphate sugar base. Well, we have a name for that. It's a nucleotide. Nucleotide makes sense because DNA is found in the nucleus. All right, so this is a nucleotide. Here's another one. And then nucleotides always have a phosphate, a sugar, and then one of the 4 nitrogenous bases. In DNA, there's A, T, G, and C. In RNA, there's A, U, G, and C. We focus on RNA in another video. So one of the things you'll notice, A's always go with T's, G's always go with C's. If you're curious as to why, I need a big base and a small base so that the size of the DNA is the same throughout. So big, small, big, small, big, small, big, small. And then the number of hydrogen bonds have to align. You'll notice that A's and T's have a couple of hydrogen bonds. G's and C's have 3. All right, so I line these things up. If you didn't know what was on this side, you would know that A goes with T, C goes with that G, G goes with that C, T goes with that A, and so forth like that. Now, if you and I don't know, that pineapple over there, there's not really a pineapple there, but if that pineapple is a plant, it has DNA, it has cells. So if it's made out of A's, T's, G's, and C's, and so am I, and so are you, why do I not look like a pineapple? It's the order of those bases. So the difference between you and me and your dog is just the order of these nucleotides. Kind of like, if you're one of my students, I've given this example several times. So apologies. The order of the bases in DNA matters, just like the order of letters, and this matters. Rat, rearrange it, tar, rearrange it, art. So different order, totally different meaning. All right, so let's go ahead and hop into a few DNA questions. All right, so this one says, a student used poster board to construct this model of a section of DNA. Which statement describes his model of DNA? A, the model's inaccurate because the base pairs are incorrect. B, the model's accurate because it contains correctly paired bases. C, the model is accurate because it shows each base splitting to form a double helix. D, the model is inaccurate because some typical bases in DNA are missing. So G with G, A with A. This is inaccurate because the base pairs are wrong. A's go with T's, G's go with C's. There are no typical bases missing in DNA. So yeah, there's just A, T, G, and C. So that one was pretty straightforward. Hopefully our STAAR test looks like that. All right. Nitrogenous bases, by the way, when you're watching these videos, I keep these in here most of the time. That just reminds me that this is actually lifted from an old STAAR test. Sometimes I pull stuff from a test bank or something that I just think is good, but it's not directly from a STAAR test. Nitrogenous bases are located on both strands of the DNA double helix. What's the significance of the nitrogenous bases? So why A, T, G, and C? What does it do? A, the number of adenines and cytosines determines the type of RNA that will be produced. B, the order of nitrogenous bases determines the order of amino acids in the protein synthesized. C, the amount of thymine and guanine in the DNA molecules determines the length of the genes. D, the type of hydrogen bonding between the nitrogenous bases determines which amino acids will be added to the peptide chain. Okay, so what do you think? Well, I think it's the order of the nitrogenous bases determines the order of amino acids in the protein synthesized. If my DNA, just for simplicity, was like CCCCC, this side would be GGGGG, then when I make my RNA and I read it in transcription and translation, then I build a protein based off of that code. But if it was instead AAAAA, TTTTT, then now that codes for different amino acids. So it's the order of bases, just like over here, rat, tar, art. It was the order of the letters. All right, let's try another one. We'll do maybe two more. Researchers combine sequences of fruit fly DNA from a gene for a particular trait with frog DNA. Cool. This mixture was heated to separate the DNA strands. So if I separate the DNA strands, what did I break? I broke the hydrogen bonds. Cooling allowed the single strands to form pairs, so they went back together. The researchers observed that some of the fruit fly DNA paired well with frog DNA. So it's kind of like this. Let's say that I've got my fruit fly DNA over here and my frog DNA over here. And let's say I heat them up so they separate. Then when I cool them off, this part sticks with this part. Doesn't that mean that they were very similar? Like, think about it. If this is all C's, this has to be all G's. If this is all C's and that's all G's, then if I unzip them and then like the C's on this side join with the G's on that side, that means that they match. So the results provide evidence that similar nucleotides are present in both fruit fly and frog DNA. Fruit flies and frogs can be made to develop some of the same physical traits. Heat can cause mutations. The replication of genetic material can occur in any temperature. Just similar nucleotides are present. That's why they're sticking together. So if that fruit fly has a whole bunch of C's right here, and so does the frog, then when I unzip this, those G's might stick to those C's. Okay, let's do one more from an old STAAR test here. This one bothered teachers because we always draw DNA and textbooks always show it going this way. And so they're like, why would you pick a diagram with DNA laying on its side? So this one says a model of a DNA molecule is shown below. You're like, all right, let's turn sideways. The arrow indicates. So there's the arrow. So the arrow indicates the bond between adjacent phosphate and deoxyribose molecules, the junction of introns and exons in the sense strand of DNA, the hydrogen bond between complementary nucleotides, the junction of a codon and a DNA triplet. So the first one, the bond between phosphate and deoxyribose. Phosphate were the round things. the sugar in DNA is deoxyribose. That's why it's called deoxyribonucleic acids. DNA is acidic. It's found in the nucleus, nucleic, and the sugar that it contains is deoxyribose. RNA is acidic. It's made in the nucleus, but the sugar that it contains is ribose. So it's ribonucleic acid, RNA. So this is not a bond between the sugar and the phosphate. So that first choice is no good. The junction of introns and exons in the sense strand of DNA, they're just messing with you, trying to sound difficult. Introns and exons, that's in RNA. So that's no good. The hydrogen bonds between complementary nucleotides. Yes, these are the A's, T's, G's, and C's along here. And we might remember from our previous picture up here that A's and T's join and C's and G's join with hydrogen bonds. All right, that'll get you through about 5 questions, and then there'll be a few extras on your Google form. Don't forget to turn that in. And yeah, thanks for listening. See ya.